Hello viewers. Today I will discuss about the glycolysis and its significance. The essential metabolic pathway of glycolysis involves oxidative breakdown of 1-glucose into 2-pyruvate with the capture of some energy as ATP and NADH. Glycolysis is important in the cell because glucose is the main source of fuel for tissues in the body. Glycolysis is a series of 10 enzymatic reactions that extract energy from glucose by splitting it into two 3-carbon molecules called pyruvate. Glycolysis is an ancient metabolic pathway, meaning that it evolved long ago and it is found in the great majority of organisms alive today. In organisms that perform cellular respiration, glycolysis is the first stage of this process. However, glycolysis doesn't require oxygen, and many anaerobic organisms that do not use oxygen, they also have this pathway. So, total reaction, the overall reaction of glycolysis which occurs in the cytoplasm is represented as glucose plus 2 NAD plus 2 ADP plus 2 inorganic phosphate, from where 2 pyruvic acid, 2 ATP, 2 NADH and 2 H plus are getting produced. So, from glucose 6-phosphate, then fructose 6-phosphate, fructose 1,6 biphosphate is produced, from where dihydroxyacetone phosphate and glyceraldehyde 3-phosphate is produced. Ultimately, this dihydroxyacetone phosphate is turned to glyceraldehyde 3-phosphate. From there, we get 1,3-biphosphoglycerate, then 3-phosphoglycerate, then 2-phosphoglycerate, then phosphoenol pyruvate, and ultimately pyruvate is getting produced. The process entails the oxidation of glucose molecules, which happens in plants, microbes and animals. All the organisms usually they use glucose except some bacteria which use actually ethanol. They prefer ethanol as sole carbon source. They are acetic acid bacteria. So these pyruvate can either be converted to carbon dioxide and water, or in the anaerobic condition they can be converted to ethanol and carbon dioxide or to lactate. In highly oxidative tissue such as heart, the production of pyruvate is very important to malate synthesis, as well as it serves as the precursor for synthesis of lactate, alanine as well as oxaloacetate. Sources of sugars. 3 main monosaccharide, they actually are served as the main sugar source, like glucose, fructose from the sucrose, galactose from the lactose diet. So they are actually fuels from diet. Glucose as dietary component can once again be retrieved from liver as well as from kidney. So all the sugars they ultimately are converted to glucose. From glycogen as well, glucose can be synthesized. Specific insight and significance. It is also called Embden-Meyerhof-Parnas Pathway. Glucose is hexose sugar, which means that it is a monosaccharide with 6 carbon atoms and 6 oxygen atoms. First carbon consists of an aldehyde group and the other 5 carbons have 1 hydroxyl group each. The hydroxyl groups allow the phosphorylation. The specific form of glucose used in glycolysis is actually glucose 6-phosphate. We will talk about the hexokinase as well as glucokinase is also present. It is a subtype of hexokinase and found in humans. Glucokinase has a high affinity for glucose and is found only in the pancreas and liver, but hexokinase is found in all cells. Biological significance. So nearly all of the energy used in the living cell, they are actually derived from this glycolytic pathway. Glucose enters in the heterotrophic cells by 2 ways. One, through the secondary active transport. So where glucose is moving against the gradient, here the SGLT1 or 2, sodium glucose transporter or symporter is used, or otherwise glucose can be entered via facilitated diffusion process with uniporter GLUT1 or other isoforms. So actually it takes place in the cytoplasm of both prokaryote as well as eukaryote, and it is the earliest metabolic pathway in the anaerobic condition. So key points, glycolysis is present in nearly all living organisms. Glucose is the main source of almost all energy used by cells. Overall, glycolysis produces 2 pyruvate molecules, a net gain of 2 ATP as well as 2 NADH molecules. So general function. In erythrocyte, lactic acid fermentation from the glucose is the sole source of ATP generation because they don't have mitochondria. But once it is matured, they have little need for the ATP. Another part of the body which relies entirely and almost on the anaerobic glycolysis is the lens of eye, which is devoid of mitochondria to prevent light scattering. Skeletal muscle usually goes for the oxidative phosphorylation after glycolysis for the generation of carbon dioxide and water. But other than these, they can also go for anaerobic glycolysis where they devoid the oxidative phosphorylation. So features. 9 metabolic intermediates between glucose and pyruvate is phosphorylated. Why? 3 functions are there. Number 1, phosphate groups are fully ionized at pH 7, so can't escape through plasma membrane as they are polar in nature. The phosphates are the essential groups to conserve energy. Ultimately, it forms ATP. Phosphate group act as binding site for the intermediates to fit at the active site of the enzyme. The reaction involved are phosphoryl transfer, phosphoryl shift, isomerization, dehydration as well as aldol cleavage. So, mechanism of glycolysis. There are 2 phases. One is investment phase and another one is payoff phase. The investment phase where the ATP is put in and the payoff phase is where the net ATP as well as NADH molecules are created. So, total 2 ATP is put in the investment phase and a total 4 ATP is made in the payoff phase. Thus, 2 net ATP is gained. The ATP created here is called via the substrate level phosphorylation. The first one, that is preparatory or investment phase, that is the energy requiring phase. So, phosphorylation of glucose happens to glucose 6-phosphate, then glucose 6-phosphate is transferred to fructose 6-phosphate, then fructose 6-phosphate by phosphofructokinase is transferred to fructose 1,6-biphosphate. Then from fructose 1,6-biphosphate, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are created. They are interchangeable. The first step that I will talk about, that is the phosphorylation of glucose. The first step in glycolysis is the conversion of D-glucose into glucose 6-phosphate. The enzyme that catalyzes the reaction is hexokinase. The glucose ring is phosphorylated. The irreversible step due to the low energy character of glucose 6-phosphate and lower stability of magnesium ATP. Phosphorylation is the process of addition of a phosphate group to a molecule derived from ATP. As a result, at this point of glycolysis, one molecule of ATP has been consumed. So, atomic magnesium is also involved to help to shield the negative charges from the phosphate groups of ATP molecules. In addition of the phosphate, it also traps inside the cell since the glucose is getting transferred to its phosphate derivative, that means glucose 6-phosphate, so they cannot pass through the cell membrane to go back to extracellular fluid. The net delta G0 is equal to minus 16.7 kilojoule per mole for this step. Next step. So, this step 1 is the phosphorylation of glucose and step 2 is isomerization of glucose 6-phosphate. So, ultimately the second reaction of glycolysis is the arrangement of glucose 6-phosphate into fructose 6-phosphate by glucose phosphate isomerase or phosphoglucoisomerase. As the name of the enzyme suggests, this reaction involves isomerization reaction. The reaction involves rearrangement of carbon-oxygen bond to transform the 6-membered ring into the 5-membered ring. So, the first carbon comes out to the ring. The net delta G0 is equal to 1.7 kilojoule per mole of this reaction. The step 3, that is phosphorylation of fructose 6-phosphate. A phosphate group is transferred from ATP to fructose 6-phosphate, producing fructose 1,6-biphosphate. This step is catalyzed by the enzyme Phosphofructokinase. It is a pivotal enzyme of glycolysis, which can be regulated to speed up or slow down. It is the pacemaker pathway of the glycolysis. It is inhibited by ATP citrate and activated via AMP. Actually, the presence of citrate inhibit to make the tetramer. It becomes in the dimeric structure. Phosphofructokinase with magnesium cofactor reacts here. A second molecule of ATP provides the phosphate group that is added to the fructose 6-phosphate molecule. As in step 1, magnesium atom is involved to help to shield the negative charges of the ATP. Here we know the delta G0 is equal to minus 18.5 kilojoule per mole. Step 4, that is cleavage of fructose 1,6 biphosphate. Fructose 1,6 biphosphate splits into two 3-carbon sugars, dihydroxyacetone phosphate DHAP and glyceraldehyde 3-phosphate GAP. They are isomers to each other, but only one glyceraldehyde 3-phosphate can directly continue through the next steps of glycolysis. Here the enzyme aldolase splits fructose 1,6 biphosphate and zinc is cofactor here. The net delta G0 is equal to 28 kilojoule per mole. Next, step 5 is isomerization of dihydroxyacetone phosphate. Now, this dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. The two molecules exist in equilibrium, but the equilibrium is pulled strongly downward as glyceraldehyde 3-phosphate is used up. Thus, all the dihydroxyacetone phosphate is eventually converted. The enzyme triose phosphate isomerase rapidly interconverts the molecules dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate is the only molecule that continues in the glycolytic pathway. As a result, all of the dihydroxyacetone phosphate molecules produced are further acted by the enzyme triose phosphate isomerase, which recognizes DHAP into GAP so it can continue in glycolysis. At this point in the glycolytic pathway, we have two 3-carbon molecules. The net delta G0 energy is 7.6 kilojoule per mole. Now, we will talk about the phase 2, that is energy releasing phase or payoff phase. Here, it is a series of reaction additional transformations where ultimately the pyruvate will be getting produced. In the process, 4 ATP molecules and 2 NADH will be produced by the sequential reactions. Step 6, oxidative phosphorylation of glyceraldehyde 3-phosphate. Here actually 3 reactions happen. One is glyceraldehyde 3-phosphate is oxidized by NAD+, and NAD+ will be converted to NADH, H+ will be produced, as well as the glyceraldehyde 3-phosphate will be phosphorylated to 1,3-biphosphoglycerate. These glyceraldehyde 3-phosphate dehydrogenase enzyme that adds an inorganic phosphate to glyceraldehyde 3-phosphate. The enzyme glyceraldehyde 3-phosphate dehydrogenase contains a structure and holds the molecule in a conformation such that the NAD plus molecule pull the hydrogen off from the gap, as well as ultimately the phosphate group is getting transferred to the gap to produce 1,3-biphosphoglycerate. The net energy change or delta G0 is equal to 6.3 kilojoule per mole. The enzyme is having thiol group, so it is inhibited by iodoacetate. The steps of reactions by the enzyme involved here is covalent binding of substrate to thiol group, then oxidation of thiohemiacetal and reduction of NAD+, then phosphorylasis of thioester. Next, step 7, that is transfer of phosphate from 1,3-diphosphoglycerate to ADP. 1,3-bi or diphosphoglycerate donates one of its phosphate group to ADP, making a molecule of ATP and turning into 3-phosphoglycerate in the process. Phosphoglycerate kinase transfers these phosphate group from 1,3-biphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. It is first molecule of ATP. Since we actually have 2 molecules of 1,3-biphosphoglycerate, we actually synthesizes 2 molecules of ATP at this step. With this synthesis of ATP, we have cancelled the first 2 molecules of ATP that was used in these first energy requiring phase. That is the first step, that is the steps in the glycolysis process. Again, we see that an atom of magnesium is involved to shield the negative charges of the phosphate group of ATP. The net energy change is minus 18.8 kilojoule per mole. The step 8, isomerization of 3-phosphoglycerate. 3-phosphoglycerate is converted to 2-phosphoglycerate. The enzyme involved is phosphoglyceromutase. It relocates the phosphate from 3-phosphoglycerate from third carbon to the second carbon. This step involves simple rearrangement process. The molecule responsible for catalyzing this reaction, that is phosphoglycerate mutase, is actually a mutase enzyme which transfers a functional group from one position to another. Sometimes 2,3-phosphoglycerate kinase can also produce 2,3-phosphoglycerate from this 3-phosphoglycerate as well, and the net energy change is 4.4 kilojoule per mole. An enzyme goes back to the original phosphorylated state. Step 9, the dehydration of 2-phosphoglycerate. 2-phosphoglycerate loses a molecule of water and it becomes phosphoenol pyruvate, which is very unstable molecule. The enzyme enolase removes 2 molecules of water from 2-phosphoglycerate to produce phosphoenol pyruvate. Actually, it happens in the pocket of the enzyme to occur the series of reaction steps. The energy involved here is 1.7 kilojoule per mole. The last step, step 10, that is transfer of phosphate from phosphoenol pyruvate to ADP. Phosphoenol pyruvate readily donates its phosphate group to ADP, making a second molecule of ATP. Okay, the enzyme involved here is pyruvate kinase. So ultimately pyruvate and ATP is getting produced. Phosphate group attached to the 2' carbon of phosphoenol pyruvate is transferred to a molecule of ADP to generate ATP here. So once again 2 ATP molecules are generated as 2 molecules of phosphoenol pyruvate is actually used here, and magnesium ATP complex is also formed here. So ultimately what happens in steps 1 and 3? 2 ATP is utilized. Step 7 and 10 produces 4 ATP. So net visible ATP produces 2. So net reaction we may say, if NADH goes to oxidative phosphorylation, glucose plus 8 ADP plus 8P plus 8H plus oxygen giving rise to 2 pyruvate plus 8 ADP plus 10 H2O. Net this reaction, that is glucose plus 6 O2 to give rise to 6 CO2 plus 6 H2O, is ultimately minus 2872 kilojoule per mole. So in case of respiration, the aerobic or anaerobic direction, it is decided by the particular circumstance of the cell. So by the aerobic respiration, it goes towards the oxidative phosphorylation, and by the anaerobic condition, it may goes towards either lactic acid fermentation or alcohol fermentation. When oxygen is available, the pyruvate is oxidized and ultimately it produces via cellular respiration to carbon dioxide as well as water, making many molecules of ATP. The fate of NADH. Glycolysis needs NAD plus to accept electrons as a part of specific reactions. So there must be the cycling back to the NADH, NAD plus as well as NADH, then to NAD plus. Otherwise, all those reactions will halt. So somehow this NAD plus will be regained back. There are some options. NADH can pass its electrons into electron transport chain, regenerating NAD plus, and as added bonus, the ATP gets made. So for one NADH, 2.5 ATPs are made. It is actually when the oxygen is present via electron transport chain or oxidative phosphorylation. But when the oxygen is absent, then these NAD plus can be regenerated either via lactic acid fermentation or via alcohol fermentation. Muscle or anaerobic glycolysis and homolactic fermentation. In muscle and lactic acid bacteria like lactobacilli streptococci, reduction of pyruvate to lactate happens by the help of enzyme lactate dehydrogenase. So many lactate dehydrogenase isoforms, isozymes are present in muscle, in heart, skeletal muscle, liver, kidney, RBC, etc. Lactate is dead end streets in metabolism. It must be converted to pyruvate as well. So what is going on? We know from glyceraldehyde 3-phosphate, 1,3-biphosphoglycerate is produced and NAD plus is converted to NADH. These NADH can be converted to NAD plus once again by the conversion of pyruvate to lactate with the help of enzyme lactate dehydrogenase. Alcoholic fermentation. In yeast, by the help of enzyme pyruvate decarboxylase, the pyruvate is getting converted to acetaldehyde. These acetaldehyde with the help of enzyme alcohol dehydrogenase ultimately produces ethanol, and from 2 NADH, 2 NAD plus can once again be regained back. So these two are the anaerobic condition where devoid of oxygen, NAD plus can be regenerated for glycolysis. Regulation of glycolysis by glucose, allosteric regulators and oxygen, enzyme induction, as well as for phosphofructokinase 1. First one, glucose. Glycolysis can be regulated by the amount of glucose available for the process, which is actually regulated primarily by two ways. Number one, regulation of the glucose uptake, regulation of the breakdown of glycogen. So when we are concerned about the glucose uptake, as I have already told, the GLUTS are the responsible glucose transporters. So there are so many isoforms of GLUTS over the plasma membrane. So actually it helps to take up the glucose inside the intracellular matrix. It can increase the uptake of glucose. Actually they are waiting within the secretory vesicle when signal comes as hormone, insulin, whatever. When the glucose concentration is increased outside, all those GLUTs are getting incorporated within the plasma membrane. There are so many isoforms. GLUT1 is found in RBC. GLUT2 is actually found in liver. GLUT3 is found in neurons. GLUT4 is found in adipocytes, and GLUT5 transporters are important to take up fructose. Another regulation is by the breakdown of the glycogen. To break down glycogen or to synthesize glycogen, two enzymes are there, glycogen synthase as well as glycogen phosphorylase. These two enzymes are actually regulated via feedback loops of the glucose or glucose 1-phosphate or allosteric regulation by metabolites as well as covalent modifications via phosphorylation, dephosphorylation, control via protein kinase A. Allosteric regulators and oxygen. We know that some processes of the glycolysis actually the irreversible steps like hexokinase, phosphofructokinase as well as pyruvate kinase. So they can be regulated via so many allosteric modulators as well as including glyceraldehyde 3-phosphate dehydrogenase. The amount of oxygen available can also regulate glycolysis. The effect is called the positive effect. It describes how the availability of oxygen diminishes the effect of glycolysis and decreased availability leads to the acceleration of glycolysis, at least initially. The mechanism responsible for this effect include the involvement of the allosteric regulators of the glycolysis. The Pasteur effect appears to mostly occur in tissues with high mitochondrial capacities such as myocytes or hepatocytes, but this effect is not universal in oxidative tissues such as pancreatic cells. Enzyme induction. Another mechanism to control glycolytic rates is transcriptional control of glycolytic enzymes. Altering the concentration of key enzymes allows the cell to change and adapt to alteration in hormonal status. For example, increasing glucose and insulin levels can increase the activity of hexokinase and pyruvate kinase, therefore increasing the production of pyruvate. Phosphofructokinase. Fructose 2,6-biphosphate is an allosteric regulator of PFK1 or phosphofructokinase 1. High levels of fructose 2,6-biphosphate increase the activity of PFK1. It is produced by phosphofructokinase 2, PFK2. PFK2 has both kinase and phosphorylase activity and can transform fructose 6-phosphate to fructose 2,6-biphosphate and vice versa. Insulin dephosphorylates PFK2 and these activates its kinase activity and increases the levels of fructose 2,6-biphosphate, which goes on to activate PFK1. Glucagon can also phosphorylate PFK2 and this activates phosphatase, which transforms fructose 2,6-biphosphate back to fructose 6-phosphate. That means this decreases fructose 2,6-biphosphate levels and decreases PFK1 activity. PFK is actually the pacemaker. It's a pivotal enzyme. Clinical significance. Pyruvate kinase deficiency is an autosomal recessive mutation disorder that causes hemolytic anemia. There is an inability to form ATP and causes cell damage. Cells become swollen and are taken up by the spleen, causing splenomegaly. Signs and symptoms include jaundice, icterus, elevated bilirubin, and splenomegaly. Arsenic poisoning also prevents ATP synthesis because arsenic takes the place of phosphate in the steps of glycolysis. So students, I have already discussed about the reactions of the glycolytic pathways, its clinical as well as biological significance, as well as the regulation of the enzyme which actually regulates the biological process of glycolysis. Thank you.